This is a recording of a small intestine, and this is jejunum. Now, we do not expect you to differentiate between the jejunum and the ileum, but we are going to show you here in this specimen the features of a jejunum, and also the next specimen the features of ileum. The ileum has one feature that we do require you to be able to identify and understand, that those are the Peyer's patches, and you can see the blue staining here, which are the lymphatic follicles that are part of the Peyer's patches in the ileum. So let's look at the jejunum first, and actually the last annotation is probably the key one to see the overall view of this specimen. No, let's see. The plica circularis. Now I'm going to move this down, and what I have in the box here is one plica circularis. It's a fold of the mucosa. Remember, the mucosa is epithelium, lamina propria, and muscularis mucosa, and the core of a plica circularis in that fold is the submucosa. So this is a way to increase the surface area for absorption and interchange between the lumen and the capillaries and the lymphatics that carry blood and nutrients away that are absorbed, as well as the lymphatics, and which transports fat. So if you zoom in on this, you can see the villi. Here's an example of one here. Here's a kind of a short villus, and some of the epithelial cells have sloughed off here. Like, this is just an artifact. They're the epithelial cells that are covering the tip of a villus, and this is the lamina propria with lots of lymphocytes, some capillaries, and then the epithelium contains many goblet cells, which you can see here. So that is the surface area increase, and the other surface area increase lies at the border, but this is getting close to the ileum, but it's not the ileum, because the further down you go in the GI tract, the higher the density of goblet cells. Now here, these arrows are pointing out 2 or 3 typical villi. Now right at the base of the villus is where there should be at a proper level of section, you would see a continuation from the lumen down into the center of this gland. These are intestinal glands, and here's the muscularis mucosa. So all these cells here in the lamina propria are either smooth muscle cells, which are the reddish stained cells here, fibroblasts, lymphocytes, plasma cells, and in the core of the villus is hard to see, and in many cases is a lymphatic as well as capillaries. So the Paneth cells are right at the base of the intestinal glands, also called the crypt of Liberkin. And I think that okay, so this annotation shows 2 crypts of Liberkin, but here's another one and another one here, etc., etc. Down here this is a vein, a small vein, venule, and here's an arteriole that's engorged with red blood cells. And of course out here, this is the connective tissue of the submucosa. And out here, now here you see the cross section of smooth muscle cells. And the inner layer of the muscularis externa is circular around. It surrounds in a circular fashion the GI tract tube, whereas the outer layer, the longitudinal muscle, runs longitudinal or parallel to the long axis of the intestine. So what kind of section is this? Well, it has to be a longitudinal section. That's why you see a fairly good length of the mucosa. So that's the way you determine that. Now I noticed over here is another vessel that's a little larger. So this vessel is now bordering on the a very small muscular or distributing artery. Okay, so I think that that's enough orientation for you to study this specimen.